Alright, in this video lesson, we begin Unit 2 on cells. Back in Regents Biology, we did quite a bit on cells. What you're going to see in this unit is a lot of review of that, along with a significant amount of new stuff, things that we didn't have to get into detail back in the Regents class. So, again, just as a review, you should have the guided notes with you, and you're using this video to complete those. And that'll maximize our time in class to be able to have discussions and activities and labs, take notes, write down questions, write down question marks on things you'd like to discuss in class so we can really focus on the harder things, the things you didn't understand compared to everything. So Unit 2, this is the first of 4, I believe it is, dealing with just cell structure and support. So first, we have these things, and when we talk about life, we have these things called the characteristics of life, things that all living things have to be able to do. And you'll see I've listed several there. Process matter and energy, process information, take in nutrients and remove waste, metabolize materials, respond to stimuli, maintain homeostasis, reproduce, transport materials, communicate. Living things must be able to do these things in order to survive. The environment is constantly changing. Organisms have to adapt. They have to respond to things. These are all characteristics that every single organism, regardless how complex it is, whether we're talking about human beings, whether we're talking about a simple bacteria, all things that these organisms need to be able to do. Now, in order to do this, if you actually really think about it, the organism has to be able to do it, but really it's the cell or the cells that perform these activities. So when you're talking about processing matter and energy really is processed inside of the cells and by the materials that the cells are actually making. We're talking about processing of information. We're talking about things in the cell that process the information, things such as DNA, RNA, ribosomes, as we'll see later on. Taking in nutrients, well, it's really the cells that are taking in those nutrients and utilizing those. Removing the waste, getting rid of those. So really, when you look at even reproduction, why is it that organisms can reproduce? Well, it's the cells that have the ability to reproduce. So what we're going to be doing in this unit is looking at the different structures and the different characteristics of cells, and really trying to get an idea of how it's the cells and the structure of the cells that really perform these different activities. So we'll start with a structure that is on the very, very outside of many, many cells, called the cytoskeleton. As it says there, the cytoskeleton is a network of different types of proteins that you'll find throughout the cytoplasm. So just inside of, generally when we're talking about cells, just inside the cell membrane, we have these different types of proteins. Different proteins called microfilaments, microtubules, and intermediate filaments. If you happen to see any one of those 3 words, just try to keep in mind that it has to do with the cytoskeleton. Inside of the cell membrane, rather, we have all of these different proteins that remind me a little bit of like Legos. Legos, you can take these things, you can use those little knobs, and you can connect them, you can disconnect them, you can reassemble them, you can assemble them, you can change the shape of them, and that's really kind of what the cytoskeleton is. If you take a look at the picture on the left, you will see you've got the nucleus, which is the bluish, purplish thing that I'll put an X on just so that we're talking about the same thing. All of these different lines that you'll actually see. And this is going to be the dark arrow that I'm drawing. This is going to be the cell membrane there. All of these other kind of lattice work type materials, that is the cell, that is the cytoskeleton itself. And so you have all these different proteins that are present there. Why are they there? Well, they're there for structural support, meaning that they help with the shape of the cell. They help with maintaining the rigidity of the cell. They help maintain the overall structure of the cell. Some cells can actually change shape, and it's because of shifting and the rearranging of this cytoskeleton that actually allows that to happen. Anchoring of organelles as well as moving of organelles. We always talk about the fact that the substances inside of the cell can move all around. Well, really it's kind of these cytoskeleton, these protein filaments, that'll actually attach to them and are able to move and direct some of these organelles to proper locations. Regulating motility, we're going to talk down below about 2 very, very important structures that are built out of the cytoskeleton, the cilia and the flagella. As well as when we get to mitosis and meiosis, we are going to see the things called spindle fibers. If you think back to Regents Biology, you might remember the spindle fibers involved in helping kind of coordinate and move the chromosomes around. Well, those are also examples of the cytoskeleton itself. So accessory proteins, They can assemble, they can disassemble, they can change the overall shape, and they are very, very important in maintaining the overall functioning of the cell itself. Now at the bottom it says two extensions of the cytoskeleton that you know, the cilia and the flagella, and it says to draw those. So if we draw a cell membrane, we'll say that these are two different cells. One on the left, we'll draw cilia. Cilia are these tiny, tiny little hairs that stick up. And it's these little hairs that have the ability to provide motility, motility meaning movement. When these little hairs are coordinated and they move, they can actually do several things. You'll see some organisms use these to take in food. So you'll see the mouth area lined with cilia. And what it does is it kind of continues to beat and pull liquid in and pulling the food in as well. Some of them are also involved in just the movement of the cell. One great example, if you think back to seventh grade, and if you happen to do the anything with pond water, there are these little organisms called paramecium. And paramecia, I'll do my best to draw them. They kind of remind me of a football, kind of, with a little dent in the side. Paramecium are covered in these little cilia, and it's a cilia that will actually allow them to move. And they're very cool because they can go forward and backwards and sideways, and it's all the coordination of these little cilia that allow that to happen. Flagella, on the other hand, off of this, you have a large single tail-like structure that comes off. Think of it kind of like if you think about like a sperm cell have the head and they have this long single tail. Well, that's what a flagella is there for. And generally, a flagella is entirely there for motility or movement, allowing the cell to move. So if you think about sperm trying to seek out the egg, that single tail that comes off of there is very important for that to happen. There's a lot of algae as well that have flagella to them, allowing them to move within the water column. So again, the cilia and the flagella, both extensions of the cytoplasm, having to do with that regulating motility or movement of the cell. But keep in mind that there's a lot of things that the cytoplasm does as well in support of the cell itself. All right, let's move on to the cell membrane itself. Feel free to pause this, take down the notes, and then it'll allow you to kind of listen better instead of having to write and listen at the same time. That might help out. So when we talk about the cell membrane, we need to go back to something that we did in Unit 1. And that thing that we need to go back to are these things called phospholipids. When we talked about the 4 different organic macromolecules, one of those organic macromolecules was the lipid. And one special example of a modified lipid is called a phospholipid. You'll see here it says to sketch a phospholipid and label it. I've given you 3 different pictures. Again, if it sort of come up on a test, it could be any one of these or something modified even from one of these. The one on the left is probably the simplest. Notice that we have the hydrophilic, or sometimes we call this the polar head. And then we have 2 either non-polar or hydrophobic tails. And just as a reminder, remember hydrophobic means that they cannot be near water. They cannot stand water. They have a phobia of water. You can think about it that way. Hydrophilic means that the head area is attracted and can interact with water. This is one of those modified triglycerides where we have, this is actually a phosphate group that makes it polar. And then we have the 2 fatty acid tails, which remember these 2 things at the bottom are fatty acids. The one in the middle shows you a little bit of the molecular structure of it. So you'll see this thing in, I think it's orange. That is the phosphate group. And that's what makes it polar, because you can see that we have some charges that are actually on there. We have an unequal distribution of the electrons. And that's actually what makes it polar area. And then here are our 2 fatty acid tails, one of them being saturated. Remember, all the carbon-carbon bonds are single. One of them being unsaturated, meaning we have a single carbon-carbon double bond that bends or puts a little kink in that. But this bottom area is hydrophobic or non-polar. The one on the right, you'll see another picture, a little bit different. This one's actually showing some of the subunit structures, I guess today, of some of the carbons. The carbons are actually present as little circles in this case. But again, all of these really show the same thing. Another word that we dealt with back in Unit 1 was this word amphipathic, and this one is spelled correctly compared to the last one. Amphipathic just describes a molecule that has both polar and nonpolar sections to it. And so the phospholipid is one example of that. It has a polar head and a nonpolar tail. And we talked about this a little bit back in Unit 1, but it creates a very, very unique situation because the cell is surrounded by water. The cytoplasm is all water. So if we have an entire half of this molecule that can't interact with water, it creates something very, very unique when we talk about the cell membrane. So at the bottom it says to draw what is called the fluid mosaic model. Obviously, I did not draw this. Also, feel free, instead of drawing it, feel free to jump onto Google, look for some pictures that might be more helpful than your drawings. We'll practice some of the drawings of these in class, but feel free to do that. Just copy it from a site and then paste it. Just make sure you understand it. Don't just take in a picture because it looks pretty. As I go through and try to find drawings, there's a lot of really, really complex ones. I try to find ones that are simple, things that we'll actually understand. So, it says sketch and label the fluid mosaic model. Let's focus first of all on the simpler one, which is the middle one. So basically what we have with the phospholipid, since half of it cannot be near water, the tails cannot be near water, and the heads can, what we create is this phospholipid bilayer. Bilayer means two layers, where we have one layer there and a second layer there. They are mirror images of each other, arranged so that the polar heads are on the outside and the polar heads are on the very, very inside. Remember, outside of the cell, we're going to have some sort of watery, liquidy fluid. On the inside of the cell, we're going to have the cytoplasm. Both of those are water-based. And so we put the heads on the very, very outside and very, very inside to ensure that those can interact. And then what we do is we sandwich, in orange here, we sandwich the tails on the very, very inside because nonpolar things can interact with nonpolar things. So that is the phospholipid bilayer. Every one of these phospholipids has the ability to shift, not only side to side. When you think about it in a three-dimensional pattern, think about this. This phospholipid can shift over here. It's almost like little ping pong balls. All of these things can shift around. And that is very, very important for the fluidity, hence the name fluid mosaic model. When you push on animal cells, when you push on cells that have cell membranes, there's flexibility to it. That is a very, very important component of that. Now, the idea of a mosaic model, if you think about mosaic art, you take some sort of tiles, colored tiles, and you arrange them to make a bigger picture. Well, when you look at the cell membrane, we have all of these different proteins, and that's what these other things are in this picture besides the phospholipids. We have all these proteins that have very, very important functions as well that we're going to talk about here in a couple minutes. These are arranged in a pattern-like system, and that's what gives it the mosaic. So if you look at this kind of from a distance, you can kind of see you've got this pattern-like structure, and that is what the fluid mosaic model deals with. Why does this arrangement occur? As it says, we've got intracellular and extracellular fluids are water-based. Only the hydrophilic or the polar heads can interact with the water. The hydrophobic tails sandwich in the middle layer away from the water. Creates a very, very stable system, and that is what every single cell membrane, regardless of whether it is bacterial, whether it is animal cells, plant cells, that is exactly what it looks like. So this is one of those similarities or commonalities between all living things. All living things have to have cells. All living cells have to have cell membranes, and the cell membranes have a common structure that looks something like that. Again, any questions, anything you understand, please make sure you make little notes to yourself inside. So let's talk about some of the proteins that are there, what some of these proteins actually do. So first of all, well, this isn't a protein. First of all, in the cell membrane, we have a lipid that is present there called cholesterol. Again, a picture I took from online. Here are the phospholipids that are connected in this phospholipid bilayer. What you're going to find is that we have these lipids in between the phospholipids, not all of them, called cholesterol. And cholesterol is a molecule that's very important for, you'll see on the right, it's a very important molecule for maintaining the fluidity of the cell membrane. We want to make sure that the cell membrane is fluid. All these different components can shift around and is flexible. Temperature can have an impact on that. When temperature gets too high, the cell membrane becomes too fluid, meaning that substances can very, very easily pass through when maybe they shouldn't be. Think about waste passing into the cell when you don't want that to happen. And so, what cholesterol can do on the higher end of the temperature is keep it a little less flexible. On the lower end, as things cool down, there's less shifting around of these phospholipids. And so, in a lower temperature, we can have less fluidity or less flexibility. What the cholesterol does is it ensures that at lower temperatures, it kind of increases the flexibility or fluidity of this membrane. So cholesterol, important for maintaining the overall fluidity in both high and in low temperatures. Next, let's move on to the channel proteins. If you think about the word channel protein, it really kind of gives you, or should give you, an idea of what you're talking about. You're talking about a protein that forms a channel in the cell membrane. Here in this picture, we have the phospholipid bilayer, and you'll see this thing right here is the channel protein. Remember, this is a protein. It's composed of amino acids. So the structure you're seeing is just kind of I guess, the big picture of what it looks like. But you got to remember that this is composed of amino acid connected to amino acid connected to amino acid. What this allows us to do is it allows polar molecules to move through the cell membrane. This area that I'm going to block off in a rectangle, remember, this is a non-polar tail area. And remember that polar things and non-polar things do not mix. So if all these little balls on the outside, these are substances that we really, really need inside of the cell. If these are polar molecules, and I'll put a P there for them, they can't just simply pass through the cell membrane because of this barrier layer. This non-polar barrier layer will not allow them through. But there's still things that we're going to need inside the cell. And so we have these specialized proteins. And these proteins extend through the entire membrane. They're called transport protein is another word that you might actually see as well. And what you'll see is that these are things that allow the polar molecules, and you see it there, to pass both into and out of the cell. So this only shows them going into the cell, but keep in mind this allows the molecules to go out of the cells as well. They can be gated. And what I mean by gated is, imagine if you would that we have this little area in the middle that kind of pinches off, and the cell has the ability to open and close it as needed. So the cell has some sort of ability, and we'll talk about this later on, how this actually happens. When needed, this channel can actually open up and allow the substances to actually come through. But when not needed, has the ability to close back up to ensure that these substances don't just continuously come in. Now here's what I want you to think about. This is a protein composed of amino acids. If you think about the amino acid arrangement within this protein, remember that we have different types of amino acids. We have polar amino acids, we have non-polar amino acids. And here's what I want you to think about. I'm going to draw a line here and draw a line here. And let's say that this is a chain of amino acids that make up part of this protein. What kind of amino acids would have to be present there? Just in those little orange lines. Hopefully, you think to yourself and you come up with non-polar. Because remember, this area is interacting with the tails. So because these areas are interacting with the tails, this has to be an entirely long chain of non-polar amino acids as well, so that they can actually interact with the tails. Now, on the vice versa, I'm going to change over to this other color, and I'm going to color in a new area. And let's say that these are chains of amino acids as well. Now, what kind of amino acids would these have to be? These are going to be the polar ones. Because remember, this is supposed to be a channel that allows polar molecules to come through. Well, if they're going to allow polar molecules to come through, and this is a watery-based area, we have to have these polar molecules lining the inside of the channel. Some questions that could come up, thinking about just the channel protein in general and relating it. So the big picture when it comes to AP biology is linking all these different units together. It's not a unit 1 test and then a unit 2 test. It's understanding how these things connect. So here's a great example of connecting unit 1, protein structure, along with unit 2, cell structure. Think about this. If we have mutations, that change amino acids, what's going to happen? If the protein is mutated, think back to conformation, and shape is very, very important. You could end up with a disorder where the channel protein doesn't work correctly. So now whatever is supposed to come into the cell. So again, trying to make all those different connections and all those different links between the units. That's the key thing to AV biology. Next, let's move on to our third type of protein, which are called the receptor proteins. If you think about the word receptor, receptors receive. And so in this case, the question is, what do they actually receive? Well, these are proteins that are also in the cell membrane that are able to receive information from outside the cell and pass it inside of the cell. These are, they have the ability to interact with these things called ligands. We're going to see this word ligands when we get to cell communication. A ligand is a signaling molecule. So this would be an example of a ligand. So imagine, if you would, that one cell releases this chemical, this ligand or the signaling molecule, and what it wants to do is tell other cells something. Well, you need some sort of way of actually like receiving the information. Because notice that this huge thing, which could be polar, can't just make it through. Not only is it too big, but again, potentially it's polar, so it can actually make it through. So how does the cell actually receive that information? It has these receptor proteins. Receptor proteins have a very, very unique shape to them. And that unique shape on the outside fits perfectly with the signaling protein, or the signaling molecule, let's say. The signaling molecule attaches to it. It's received. It passes the information into the cell, whatever that information is. Maybe it says, hey, I need you to multiply by mitosis. I need you to release a certain chemical such as insulin. I need you to release, maybe an enzyme of some other sort. Um, that's what the purpose of the receptor protein is actually to do. Now, one interesting thing, we have these receptor proteins. Some viruses can actually hijack these receptor proteins and actually utilize them to enter into a cell sometimes, which is kind of interesting. So if we have this area that can receive things, if the virus has been modified to fit perfectly into that, it can actually make use of that ability as well. Last type of protein that we have in the cell membrane are what are called marker proteins. In marker proteins, I didn't really find a picture that only has um, only the marker proteins themselves. The ones that we're looking at, I'm going to cross out in, I'll find this dark blue. I'm going to cross out in the dark blue. We don't want this one. We don't want this one. We don't want this one. You can see that's a channel protein. The ones that we're focusing on, and I'll circle these in the orange, are these ones that have these side chains that are sticking out the outside. So when you draw these, those are the ones you actually want to draw. These are examples of marker proteins. They're sometimes called glycoproteins because what it is it is a protein. That's what the bluish, grayish, greenish, purplish, I don't know what it is. That's what that is the protein part. And then the glyco part has to do with a carbohydrate. And so these are carbohydrate side chains. These are sugar side chains that stick off. They stick off generally on the outside because these are responsible for what we call, these are markers for cell identification, sometimes called cell to cell recognition. Now, what do I mean by cell to cell recognition? Well, cells have to be able to identify other types of cells within the body. For example, your white blood cells have to be able to identify what cells should be there and what cells shouldn't be there. And so when we talk about cell-to-cell identification, cell-to-cell recognition, what we're looking at is we're looking at identifying the cell type, meaning is this a blood cell? Is this a white blood cell? Is this a red blood cell? Is this a skin cell? Is this a certain type of skin cell? A certain type of nerve cell. These identification markers on the outside, these marker proteins, allow the body to know exactly what type of cell it is, and the cell source. What I mean by cell source is one of two things. It's either itself or a foreign cell. Your cells are marked with marker proteins to say that they're your cells. These are cells that are supposed to be here. They're sourced from us, and these are the ones that are supposed to be in the body. Any other cells that try to get in are going to have different marker proteins on them. What the body has the ability to do is recognize these other cells, these foreign cells, and then basically destroy them. This is what allows the body to fight off bacteria, to fight off viruses, to fight off allergens, to fight off things that shouldn't be there. Because when the body looks at all the different cells in the body, or when the immune system looks at all the different cells in the body, anything that's not supposed to be there, anything that has a different marker protein, is going to destroy. Now, there's a thing up top that says, how does this apply to organ transplants? Everything just went. So, when an organ transplant occurs, you get put on a donor list. And that donor list is there because you just can't take any organ and put it into a human body. That organ has to have marker proteins that are close enough to your own so that the body doesn't recognize it as a foreign entity. You get put on a list. There are these specialized proteins, these marker proteins that are sequenced in your body to determine exactly what marker proteins you have. And then why you're put on this list is because there are lots of organs that are donated, but they have to find the one that matches closest to yours. And so that's why sometimes you're on this list for months, sometimes for years, and sometimes you never find an organ, unfortunately, to match. If you were to take just any organ and put it in the body, even though it's there to help you survive, to help you live, it's going to it's going to be destroyed by the immune system. And so marker proteins are very, very important things. Again, you'll see they're the ones that are circled in the orange. And that includes all the different types of membrane proteins that you'll see in the cell membrane, again, fluid mosaic model. Now, in addition to what we just gave up top, there are some other functions of membrane proteins as well. And so intercellular joinings, proteins are also involved in joining cells to other cells. We're going to see some examples of those at the end of this unit, or at the end of this part of the unit, rather. Enzymatic activity, some proteins actually function as enzymes, able to convert molecules into other molecules, whether it's combining them together or breaking them down. So we actually have some enzymatic activity because of proteins that are embedded in the cell membrane. Here we talked about transport. We have in Unit 2, we have a second part that'll be dealing with types of transport, so we'll see some of these proteins again. There's a cell-to-cell recognition. Notice again, we have this glycoprotein, this carbohydrate side chain is sticking up. Anchoring and attachment. For example, we need proteins in order for the cytoskeleton to be anchored to it. And so you'll have these specialized proteins that allow those to be anchored. Signal transduction is this whole communication thing. We have an entire unit on cell communication and signal transduction and how that actually works. So these are the receptor proteins receiving the information. How do they receive it? How do they pass it in? But that's another important function. And then again, we have some other ones, attachment to the cytoskeleton, just talked about. Endo and exocytosis, we'll talk about those in another part of this unit as well. And then again, the cell-to-cell communication. So lots and lots of functions of the proteins that you find in the cell membrane. Knowing the structure of the cell membrane, we now need to look at some of the functions of it. There's a big long list. Again, I would suggest you pause it. Take down what you need to from this. And maybe it's not copying down everything. Maybe it's just copying down the things that will help you understand it. The cell membrane is a selective barrier. It's a selective barrier that has what's called selective permeability, has the ability to control what passes in and out, not only because of the phospholipid bilayer, where we have the polar and the non-polar barrier on the inside, but also because of things such as channel proteins. Channel proteins that can be closed, can be gated, allowing certain things in and out. So one of the biggest things you'll see, and hopefully remember from Regents biology, that the cell membrane is very, very selective in what allows in and out. Cell identification we talked about, not only the type of cell, but also the source of the cell. Remember, the source of the cell, we're talking about either self cells or foreign cells. We just talked about with the marker proteins. Signal transduction and communication. It is a very, very important boundary layer. It allows the transport of necessary materials in and out. You don't want to allow everything into the cell. You want to be somewhat selective. This is an important one here. Because of the ability to be selective, What the cell membrane can do is it can actually maintain unique intracellular environmental conditions. Sometimes the inside of cells have a different pH. They have a different concentration of proteins, of salts, of ions. And that can't happen unless we have a selectively permeable cell membrane to it. Intercellular joining, and we'll talk about that here in a little bit. Enzymatic activity, attachment to the cytoskeleton, maintaining fluidity, flexibility of the cell. The reason that you can push on your arm and it is flexible and not a rigid structure is because of the fluidity and the flexibility of all of these different cell membranes in all of the cells that you find. So those a relatively complete list of what the cell membrane does. A super, super important structure on the outside of every single cell. Does a lot for the cell. Right, so we move on. A few other structures. Again, what we're really focusing on here is what's on the outside of the cells for now. So let's talk about the cell wall. So we said that the cell membrane is around every single cell, and that is correct. In some cells, there's an additional structure outside of that cell membrane called the cell wall. We're mainly talking about plant cells, cells of fungi, and some bacteria, many bacteria, but bacteria as well. And so what the cell wall is a cross that says lined. That should actually be the word. See if I can change it. Linked. So a cross-linked network of structural polysaccharides. Remember that a polysaccharide is another name for a complex carbohydrate. So in plants, cell walls are made of a substance called cellulose. Cellulose is a carbohydrate, and it's one that we have a very, very hard time breaking down. We don't have the correct enzymes to actually break it down, so that's why when you eat corn and other vegetables, you can chew it, but it doesn't really break it down all that well. Enzymatically, it doesn't break it down. You can mechanically break it down, but that's it. In fungi, their cell walls are composed of another polysaccharide or carbohydrate called chitin. And in bacteria have a cell wall composed of peptidoglycan. And so in the picture down in the bottom left, you will see there is a typical plant cell. Here is the cell membrane here. And on the very, very outside of that is this very, very rigid structure called the cell wall. The picture on the left shows you, here's cellulose. Cellulose is composed of glucose molecules. Remember, glucose is a hexose, or a sugar that has 6 sides to it, 6 carbons. And so notice that we have this big, long chain, cross-linked chain of these glucoses, and that's what forms the cell wall. Location outside the cell membrane. Functions. Structure is the big thing. Structure and shape. The cell membrane is flexible. The cell wall is not. And so when we're talking about these plants that don't have a skeletal system, yet they're able to grow upright. And some of these trees are able to grow hundreds and hundreds of feet up into the air. Well, it's really the cell wall that provides for this structural support. Very, very rigid structure. It's able to withstand gravity. It's able to withstand turgor pressure. Turgor pressure is when we have a lot of water inside of the cell. That water puts pressure on the outside of the cell. Kind of think about like a water balloon. The water inside of the balloon is putting pressure on the outside of the balloon itself. Well, that's what happens in these cells as well. And so the cell wall is able to maintain or withstand that. Protection mainly from physical damage to it. And remember that the cell wall is very permeable. It is not selective at all. And so that's why even in a plant cell, we still need a cell membrane inside of that to serve as this selectively permeable structure. Not present in animal cells, only present in plants, fungi, and bacteria. Finally, let's end with one of those functions that I said is important when it comes to the proteins, which is the cell-to-cell junctions. Intercellular junctions are proteins that connect different cells together. Cells just aren't kind of hanging out by themselves. They are united or joined together in some form so that we have this tissue. Remember that a group of cells that are held together is called a tissue. Functions of them, they can create channels for transport, communication, and coordination. They can provide waterproofing seals, and they can also anchor cells to base layers. And we'll talk about some of the specific examples. You'll notice over here in the picture, we're going to talk down below about these things called tight junctions, which is one example. We're going to talk about these things called desmosomes, which is another example. We're going to talk about these things called gap junctions as well. And then we have one more different type, which is called the plasmodesmata, which is only present in plant cells. What you're looking at here is you're looking at cell number 1, you're looking at cell number 2, you're looking at cell number 3. And so what these are showing you is they're showing you how these things can be joined together. Because I'll go to the dark blue area. This represents a cell membrane. This represents a cell membrane. And so these sections are showing you how we can actually combine those together, how we can join those together. So let's talk first of all about only the plants. Plants have a very, very special type of cell-to-cell junction that is only found in plants, and that is called the plasmodesmata. An easy way to remember it, plasmodesmata, only present in plants. The first two letters are exactly the same. Generally, what we think about is we think about a cell, a plant cell, a plant cell, a plant cell. You have all these cell walls. All these cell walls are kind of like glued together. Well, there's something very interesting that happens between these cells, and that is the function or the structure rather, or the presence of these plasmodesmata, and they're basically like channels. So you notice there is one right here that's being shown. In between many, many plant cells, there are actually these channels where the cell wall has kind of been, I don't want to call it broken, but it's been interrupted. So you actually have this channel there that allows the cytoplasm, which is the yellow substance, of one to be directly connected to the cytoplasm of another. And so these open channels allow for materials to be very, very quickly moved through. You now do not have to send the substance out of the cell where potentially it could get lost. You send it right from cell to cell. Very quick, very efficient. Generally, we're talking about probably diffusion. It might be some sort of carrier protein that moves these substances as well. But generally, we're talking about diffusion, where one cell can produce something, cause it to flow to another cell, and just continue from cell to cell until it gets to the location it's supposed to be. Now, moving on to the animal cells. Animal cells have several different ones. So first of all, in the functions, you'll see in the bottom right-hand corner, these are just the pictures. So here is one cell membrane of one cell. Here's another cell membrane of another cell, again in the orange. The first type of cell-to-cell junction that we have in animal cells is called the gap junction. And the gap junction is really kind of the same thing as what a plasmodesmata is in the plant cell. The gap junction is a channel between two animal cells. Again, doing the same thing, it allows for substances to move from animal cell to animal cell. In cardiac muscle, for example, think about how coordinated you have to have this. You've got to have this coordinated contraction of cardiac muscle to make sure that the blood is pumped efficiently. And so these gap junctions are very, very important there to ensure that the communication in order to contract is actually coordinated properly. Next, we have these things called desmosomes. Desmosomes, again, here's one cell membrane, here's another cell membrane. Kind of reminds me of like Velcro. It allows for one group of cells to be attached to oftentimes a base group of cells. So if you think about the skin, you have an upper layer of skin, you've got a lower layer of skin. These desmosomes allow for those different cells to actually attach together and withstand, as you're going to see, it withstands some of the tension that's there, some of the stressors that are there. And so in cells such as cardiac, skin, the linings of organs, desmosomes are very important for withstanding and maintaining integrity when there's a lot of physical stressors that are put on this. Think about the heart constantly beating. Think about the skin constantly being moved and hit with things. Very important for that. Finally, we have what are called the tight junctions. Tight junctions are sometimes thought of like rivets. If you think about a rivet like on jeans, these are things that can seal, connect one cell to another cell and seal them as well. And so these are waterproofing junctions. They're there to make sure that nothing gets between the cells. The only way for substances to move would be actually to go through the cell membrane. So by sealing these, you can make sure that you're controlling what can actually go into and out of the cells. Gap junctions are very important for things such as the small intestine, where you only want to absorb certain nutrients that you actually want. You don't want everything to come flooding into the circulatory system, into the bloodstream. And so these are very important for that. So in summary, There are the functions for those different types of proteins. We got the gap junctions, which are the open channels allowing materials to move between, generally by diffusion. We've got the tight junctions, which are the cell-to-cell waterproofing, um, ensuring that only certain things pass in and out if they want to. So you'll see important for the skin, organs, blood vessels, cavities. And then there are the desmosomes, which are the rivets that anchor some cells to base layers where there is significant stress. And that is the end of the first part of Unit 2. An introduction to some of the external structures of plant cells. What we're going to be cells in general, rather. So what we'll be doing is we'll be continuing to look inside of the cell and start to look at some of these other structures that are present inside of the cell, what they do, and how they help.